membranes are selectively permeable. What does this indicate? This means they allow the movement of only certain substance freely across them. Okay. Here you can appreciate that some substances enter freely whereas some are made to enter forcefully. The substances that enter freely are passive transport and those that enter forcefully are active transport. Basically, the structure of our cell membrane is well suited for the transport of substances in and out of the cell. Now, let me ask you something. What exactly is the composition of our cell membrane? It's mostly lipids and proteins. The next question is, do they play any role in transport across the membrane? Certainly yes. These lipids and protein of the cell membrane itself play a major role in the transport of various substances between the extracellular fluid and intracellular fluid. We will discuss about this in detail in the coming sessions. Coming back to the two ways of transport across a membrane. First, we will deal with passive transport. After that, we will move on to active transport. What is passive transport? Passive transport is the movement of a molecule down a concentration gradient from an area of its high concentration to an area of its low concentration. It is also known as diffusion or downhill movement. From the name itself, this process is passive. That is, it requires no input of any additional energy. The concentration gradient alone is enough to drive the process. It's just like swimming in the direction of water flow in a river. Getting into more details, diffusion is of two types: simple diffusion and facilitated diffusion. Simple diffusion of substances occur either through the lipid layer or protein layer of the cell membrane, and facilitated diffusion occurs with the help of the carrier protein of the cell membrane. So, the diffusion can be discussed under three headings: simple diffusion through lipid layer, simple diffusion through protein layer, and facilitated or carrier mediated diffusion. The lipid layer of our cell membrane is permeable only to lipid soluble substances like oxygen, carbon dioxide and alcohol, and therefore the diffusion of these substances through this lipid layer is directly proportional to the solubility of the substance in lipids. Whereas the protein layer of the cell membrane is in turn permeable to water-soluble substances. Mainly, electrolyte diffuses through this protein layer. When we zoom in, we can appreciate that throughout the lipid layer of a cell membrane, there are some pores in which integral protein molecules of protein layer invaginate from either surface of the cell membrane. These pores form the channels for the diffusion of water, electrolytes and other substances that cannot pass through the lipid layer. As the channels are lined by protein molecules, these are called protein channels. A characteristic feature of the protein channels is selective permeability. That is, each channel can permit only one type of ion to pass through it. And according to that, the channels are named after the ions which diffuses through these channels, such as sodium channels, potassium channels, etc. Another peculiar characteristic is some of these protein channels are continuously open. Meanwhile, most of them are always closed. Continuously open channels are called ungated channels. Closed channels are called gated channels. These channels in turn are open only when required. coming on to facilitated or carrier-mediated diffusion. It's a type of diffusion by which the water-soluble substances having larger molecule are transported through the cell membrane with the help of a carrier protein. Don't confuse this with the earlier one. There it was protein channels and here it is the carriers. And through this process, the substances are transported across the cell membrane faster than the transport by simple diffusion. Let's understand this with an example. Glucose and amino acids are transported by this facilitated diffusion. However, these glucose or amino acid molecules cannot diffuse through the channels because the diameter of these molecules is larger than the diameter of the channels. So, the molecules of these substances bind with the carrier protein. And followed by this, there occurs some conformational change in the carrier protein. As a result of these changes, the molecule reaches the other side of the cell membrane. Now, we have an overall picture of passive transport, right? So, what will be an active transport then? Active transport is the movement of molecules from an area of lower concentration to high concentration, that is up or against the concentration gradient via specialized membrane proteins. As this is against the concentration gradient, it cannot occur passively and requires energy. Now the question is, what is the source of energy for these cells? It's our ATP, of course. So the energy is provided by the breakdown of this ATP. When coming to the specialized membrane proteins, that's our carrier protein which are involved in active transport. They are of basically three types: unipod, sympod and antipod. Unipod, like the name says, they carry only one substance in a single direction. The sympod in turn is a carrier protein that carries two substances at a time and is in the same direction, whereas antipod transport two substances at the same time but in the opposite direction. Okay, now coming to the mechanism of active transport. It can be explained very vaguely like this. When a substance that needs to be transported across the cell membrane comes near the cell, it combines with these carrier protein of the cell membrane and forms a substance protein complex. This complex then moves towards the inner surface of the cell membrane. Sequentially, the substance is released from the carrier proteins. After this, the same carrier protein moves back to the outer surface of the cell membrane to transport another molecule of the substance. Okay, well, like in the case of passive transport, active transport also have different types: primary active transport and secondary active transport. Primary transport is the type of transport mechanism in which energy is utilized directly from the breakdown of ATP. An example for active transport will be our transport of sodium and potassium by the sodium-potassium pump. Sodium and potassium ions are transported across the cell membrane using this carrier protein called sodium-potassium pump. Precisely, what does this pump do? This pump in turn transports sodium from inside to outside the cell and potassium from outside to inside the cell. Well, this pump is present in almost all cells of the body. So, what happens over here? K, that represents our potassium, finally reaches the intracellular fluid. Now, taking K from potassium and I from intracellular fluid, Ki. Okay? Let me ask you something. Do you know any chocolate that starts with K and I? For me, it's Kit Kat. So remember this Kit Kat to connect with the direction of the movement of potassium. Coming back to our sodium-potassium pump. When talking about its main function, this pump is responsible for distribution of sodium and potassium ion across the cell membrane and the development of resting membrane potential. Looking at this diagram, see over here is a carrier protein. In stage 1, 3 Na plus from intracellular fluid and 2 K plus from extracellular fluid bind with C. Coming to stage 2, binding of Na plus and K plus to C activate the enzyme ATPase. The ATPase causes the breakdown of ATP into adenosine diphosphate ADP with the release of one high energy phosphate. And finally in stage 3, the conformational change occurs in C followed by the release of Na plus into ECF and K plus into ICF. Another example of primary transport is the hydrogen pump that helps in the transport of hydrogen ion across the membrane. Here also, the source of energy is ATP. And these pumps are mainly seen in two different organs of our body. One is stomach, involving in HCl formation. Another one is kidney, involving in the secretion of hydrogen ions from the blood into the urine. Moving on to the secondary active transport. Secondary active transport is a transport of substance along with sodium ion using a common carrier protein. When sodium is transported by a carrier protein, along with it, another substance is also transported by this same protein simultaneously, either in the same direction of sodium movement or in the opposite direction. And based on the direction, secondary active transport is of two types: co-transport and counter-transport. Sodium co-transport is a process in which, along with sodium, another substance is transported by a carrier protein called symport. Same direction, right? Here also, energy for the movement of sodium is obtained by the breakdown of ATP. And this energy released by the movement of sodium is utilized for the movement of another substance. Examples of substances that are carried by sodium co-transport are glucose, amino acid, chloride, iodine, iron, etc. In the case of sodium transport of glucose, one sodium ion and one glucose molecule from the ECF binds with the respective receptor site of carrier protein of our cell membrane. Now, the carrier protein is activated, causing some conformational change in the carrier protein so that sodium and glucose are released into the cell. Whereas, in case of counter-transport, sodium counter-transport is a process by which the substances are transported across the cell membrane in exchange for sodium ions by a carrier protein called antipod. The point to remember is, in co-transport, it was sympod. Here, it is antipod. Movement is in the opposite direction. An example of counter-transport system is sodium-calcium counter-transport. In this, sodium and calcium move in opposite direction with the help of a carrier protein. Okay. This type of transport is present in almost all the cells. Okay. That is all about the movement of substance across the cell membrane